Thank you for your presentation. Thank you for being here. So today I will try to give you a brief overview of what bioprinting is and how it can be used for the fabrication of in vitro models, also with some highlights on what we do in our University of Pisa. So first of all, bioprinting is part of the biggest research field of tissue engineering. So the aim of tissue engineering is to recreate artificially a tissue or an organ in order to be restored or to be replaced by combining different principles of engineering and life science. And the same is for regenerative medicine. Both can be used, both have the same aim. Which is the paradigm of both tissue engineering and regenerative medicine, is to regenerate, to substitute a tissue or an organ with a scaffold. A scaffold is a sort of architecture that can be colonized with cells. So by adding cells and growth factors to let cells maturate and differentiate, we can obtain artificially a construct that can be then further implanted in the human. Of course, a scaffold should have many properties in order to be compatible with the human. First of all, should be biocompatible, and then should mimic all the properties that are necessary to create a functional tissue. So, for example, should have the right mechanical properties, should give the right biochemical stimuli. In this way, the cells that are inside the scaffold can receive the proper cues in order to differentiate towards the right tissue. The most important thing is that a scaffold should be three-dimensional, because cells are used to live in a three-dimensional environment. So the stimuli that receive should be those of a three-dimensional space. And there are different techniques that can be used to create a three-dimensional structure. Today we will focus not on conventional fabrication techniques, I will not talk about them, but if we want to talk about bioprinting, we should first talk about additive manufacturing in order to understand what bioprinting is. So additive manufacturing is a group of technologies that basically aims to build a three-dimensional object of any complex geometry by starting from a digital model that can then be processed using software and a computer, and can then be sent to a machine, a 3D printer, in order to be produced layer by layer. So in this way we can virtually create objects of any complexity. The same is for tissues that are extremely complex objects. So if we talk about additive manufacturing in the biomedical field, there are a lot of applications. As you can see in this slide, these are only few of the applications that can be found in the biomedical field. In the upper corner on the left, this is an example of bioprinting, because it is an ear model that was realized in our lab in Pisa. But the additive manufacturing is also fabricating guides for dentistry, for example, as you can see in the top middle part, producing prostheses with metal for the orthopedic field. Or also, as you can see here, producing phantoms for surgeons, because we have the possibility with these technologies to mix different materials, different colors, different mechanical properties, so surgeons can be guided in their surgical operations with very realistic phantoms. So to sum up, thanks to the additive manufacturing, we have many advantages, because we can create very complex objects with combining different materials. We can create customized parts. Of course, we have also some disadvantages, because all these technologies are time consuming. Using a 3D printer to fabricate an object can take up to several hours. And also, these technologies can be more expensive than traditional techniques for creating scaffolds, for example. When we combine additive manufacturing techniques with biomaterials, so materials that are compatible with human cells, human tissues, we can talk about bioprinting. So basically we have our 3D printer, we use a specific material that can be with or without cells. We will learn what does it mean. And when we use this technology to create a scaffold that is cellularized or not, we can talk about bioprinting. Thanks to the bioprinting technology, so we are able to manipulate cells because we have lots of freedom in the fabrication's parameters. For example, the temperature. We can work at a very precise temperature because we can work at the physiological temperature, 37 degrees. But of course, we also have some disadvantages because we have to be sure that we are working in a sterile environment. And also when we work with cells, this can be damaged during the deposition, for example, depending on the technology used. Which are the most used technologies? The most diffused for sure is the extrusion bioprinting. In this case, also we have a reservoir, basically a syringe that contains the material. The material is typically an hydrogel, so it's water-based hydrogel, and has the same properties of, just to make an example, of hair gel. So this material is pushed toward a nozzle, and the actuation can be it's typically a mechanical actuation that can be driven by a pneumatic actuation, a piston, or a screw-driven actuation. And since the cells pass through a very small nozzle that is around 100 to 400 microns, this is the crucial part because here cells can be damaged. So this technology can be used for bioprinting, but we have a certain amount of cells that will die during the process. So the maturation of the construct will be for sure longer. Another used technology is the inkjet bioprinting. So in this case, we have a cartridge that contains a liquid suspension with cells, and we can eject small drops of material in the order of picoliters. So in this case, the resolution is much higher because we can process small amount of material. We can precisely locate in our scaffold in order to, for example, deposit different cells in different locations and obtain in this way a tissue that contains different cells, for example. And then the last one is the so-called LIFT, that stands for liquid induced transfer forward. In this case, the deposition of the material is induced by the application of a light source. So we have a laser that hits on a surface containing an hydrogel, also in this case, and releases, also in this case, small drops of material. So here we have also a higher resolution than the previous one, resolution of a single cell. So we are talking about 10, 20 microns. And in this case, the vitality of the cells is very high because cells do not have to pass through a nozzle. So there are no damages on the membrane of the cells. So this is the technology where the resolution is higher. The disadvantage of this technology is that there are not so much material that can be used. So the extrusion approach is the one that allows us to test different materials. There is a bigger amount of materials that can be processed. Instead, in this case, not so many materials can be used. Which is the advantage of having all these different technologies is that we can combine them to obtain multi-material and multi-scale scaffolds. So we can create tissue models where different technologies can be combined, and in this way we can replicate different scales of tissues. For example, if we think about osteochondral tissue, here we have the cartilage, then the bone. We have a transition phase between these tissues that can be easily replicated combining different technologies, because, for example, we can use the extrusion technology to have the precision in the order of micron. Then we can use in the order of hundreds of microns. Then we can use lift, for example, to have the precision in the tens of microns. And also these technologies can be combined with different technologies that are not part of the bioprinting field. For example, electrospinning. I will present you an example in the next slides where a scaffold was fabricated by combining the extrusion bioprinting with electrospinning. Electrospinning is a technology where a sheet of fibers is created by applying an electric field to a reservoir of material. And in this case, we can combine the nanoscale of the electrospinning with the microscale of bioprinting. I will skip this one. Which is the major challenge? The major challenge is, as I said before, is to find the right material for our bioprinting process. And especially if you think about the extrusion approach that is the most diffused, the difficulties is to find a material that is able to maintain its shape once printed. If you think that we are processing hydrogels, these usually have not the proper mechanical properties to maintain their shape. So we have to think about different approaches to be used to replicate the complexity of tissues. And actually different approaches were tried in the literature. I will show you some of them. So the first is called bioplotting. So in this case, we have our reservoir, our syringe with our hydrogel. And if we want to print a structure like this with interconnected pores, the only way is to, not the only way, one of the possible ways to print this structure into a supporting buff. So we have another hydrogel that is used as a support matrix. And our syringe moves inside this buff, creating our geometry. Then this can be polymerized in different ways, typically in a chemical way. And once it is ready, it has the right mechanical properties, this can be removed from the buff. So we can obtain a complex structure with internal cavities using this sacrificial material. A second approach is to use different printheads. So we have different reservoir with different hydrogels. Also in this case, so we have the material of the scaffold and other sacrificial materials that are only used as a support, but layer by layer. So for each layer we can print a path that is of our model and a path that is of the support matrix. And then the last approach is the core shell approach. So in this case we have a coaxial needle, so we are extruding in the same moment two different materials. One is the inner core and one is the outer core. You can see it here. So we have a red part inside and a gray part outside. So you have to imagine that we have a coaxial needle and we can extrude in the same moment two different materials. In this way we can, for example, obtain a cavity inside this material. If you want to replicate, for example, vessels inside a structure, so to replicate the vascularization, we can print the material for the vessel wall on the outside and then an inner core of sacrificial material that can be washed away when the outer core is correctly polymerized. So in this way we can print cavities inside a scaffold, for example. What we do in Pisa. Now I will show you some examples of bioprinting applications to create advanced in vitro models. So all this was a brief overview, and all these technologies have been used in the literature to create different in vitro models. In Pisa, we focused our research in the last few years on different in vitro models. The first one that I will go to present is a vascularized bone in vitro model. So in this case, we exploited the extrusion bioprinting approach. So we have a reservoir, a syringe containing a biomaterial based on gelatin, containing a mineral part that is nanohydroxyapatite, that is the same mineral part of the bone. These biomaterials were mixed and were printed into a supporting buff. That was one of the first example of the approaches to create complex structures. So we created this scaffold with internal pores without cells in a first moment. So we obtained this scaffold. This was cellularized in a second moment. So it was produced not in sterile conditions, it was sterilized in a second moment. And then cells were added inside these scaffolds. In particular, we added two different types of cells, mesenchymal stem cells to let them differentiate into the bone phase, and endothelial cells in order to let them differentiate into a capillary network. And actually, what happened is that after 14 days, a capillary network appeared inside the structure. So having the proper stimuli from the scaffold, this in vitro model proved to be efficient for recreating avascularized in vitro model. Last example that I will show you is the fabrication of a tendon scaffold. In this case, we combined different technologies, the extrusion technology for replicating in the bone phase that was combined with the electrospinning approach. These two parts were partially overlapped in order to obtain a transition phase between the cartilage and the bone tissue. And the behavior was very similar in terms of mechanical properties to the real tendon. So this was my last slide, and I thank you for your attention.